So I'm glad to take this opportunity to talk about my recent findings on the role of polyunsaturated fatty acid molecules and p38 MAP kinase pathway in bridging the gap between the metabolic reprogramming and the cytoprotective response during dietary restriction. So, food, as we all know, is the basic necessity and the integral part of our lives. And the information about food is relayed to rest of the organism through various nutrient sensing pathways like insulin, IGF signaling, TOR signaling, AMP kinase, twins, and the pharmacological interventions to these signaling pathways has led to extension in the lifespan. So basically deriving the proper nutrition out of food is the most effective and least costliest ways to reduce the burden of many diseases like obesity. India falls in the category of top 10 fattest countries in the world. So just by reducing the amount of calories intake without getting malnourished can actually reduce the metabolic disorders or complications and can extend the lifespan. For example, we have a very well-known study in the non-human primates, wherein dietary restriction has shown to better the health span and lifespan of these rhesus monkeys. Dietary restriction is the only non-invasive intervention that has led to actually extension in the lifespan in most of the model organisms tested. But in our lab, we use C. elegans or the worms as the model organism to study the biology of DR. Because of the attributes like short lifespan, cheap maintenance, easy storage, high fecundity, these are the certain attributes which has made the C. elegans to be best suited for learning about lifespans and the aging process. The powerful genetics like the systemic RNAi and CRISPR nowadays has actually enabled us to generate transgenic lines, and more so it provides us with a whole organism perspective in vivo. So using these wonder worms, we are actually we have established that eating in a right quantity as well as in right amount is equally important to provide the beneficial effects. So in 2014, we published in Aging Cell that dietary restriction-like condition inside the organism led to increase in the lifespan through coupling of metabolic reprogramming and cytoprotective response. And recently in 2018, we published in PLOS Genetics that different types of E. coli feed given to the worms like OP50 and ST115, they were actually sensed differentially by a gene called as FLR4, making a gene diet pair, which led to extension in the lifespan through the utilization of p38 MAP kinase pathway. This supports that not only the food quantity but also food quality matters to give the beneficial effects. Now, to have a deeper understanding of how food quantity matters, we actually exploited a novel gene which is called as DRL1. So this DRL1 is basically a MEP3-like kinase in C. elegans, which on knockdown in the hypodermis and in the muscles led to a metabolic shift towards higher fatty acid oxidation through utilization of this nuclear hormone receptor 49, which actually led to increase in the beta oxidation genes like ACH2, ECH9, etc., leading to less of fat storage, which is the characteristic phenotype of a dietary restricted worm, and also low ROS. Then this somehow led to the activation of cytoprotective response by enabling the increased activation of xenobiotic detoxification genes via FA4 FOXA transcription factor or NHR8 nuclear hormone receptor. Both the coupling of this metabolic reprogramming to cytoprotective response led to extension in the lifespan. However, the link connecting the two is still the black area, which needs to be delineated to have a deeper or better understanding of what is happening downstream to DRL1. So for that, what we did was that we did a targeted screening of various longevity signaling pathways, and what we came up with was the p38 MAP kinase pathway. So p38 MAP kinase pathway is the only innate immunity pathway happening in C. elegans. So this is a typical lifespan curve where number of days is on x-axis and percentage alive on the y-axis. So knockdown of DRL1, as I mentioned, led to extension in the lifespan with respect vis-a-vis of that of control. However, knocking down or mutating any of the component of this p38 MAP kinase pathway, be it NSY1, which is a MAP kinase, SEC1, which is a MAP kinase, or the downstream molecule PMK1, which is a p38 MAP kinase. So any of the component of p38 when was abrogated did not actually lead to extension in the lifespan by DRL1, showing its requirement. We also checked the requirement of p38 in other DR paradigms, E2 being the standard DR model, wherein there is extension in the lifespan, but when p38 was mutated, the extension could not happen. We also utilized other non-genetic DR paradigm, which was by utilization of different dilutions of bacteria, which led to extension in the lifespan at a particular dilution, and then going back because of the starvation that kicks in. So this gives a typical bell-shaped curve in a wild-type scenario. But when a gene is required, for example, here in SEC1, p38, if it is required, then there is no extension, and it feels like there is a flat curve, showing that p38 is also required in the non-genetic DR regime, BDR. We also utilized another non-genetic DR regime by feeding the worms with the less metabolizable glucose counterpart, which is 2-deoxyglucose. But herein, p38 was not required. Now, the specificity lies in the requirement of p38 MAP kinase pathway is specific to DR, because when p38 was mutated in any other pathway, like in the insulin signaling pathway, then the extension was still there, and the other MAP kinase pathway, like JUNT pathway, it was also not required in the DRL1. So it was specific of this p38 MAP kinase pathway, which was required under dietary restriction. So the important question now was where to place this p38 MAP kinase pathway. What role is it playing? Is it required for the metabolic reprogramming, or it was required for the cytoprotective response? So for that, we did some experiment to look into the metabolic changes under dietary restriction. So we first looked into the fat storage by the oil redo staining protocol, and the characteristic feature of dietary restricted worm is the depleted fat storage, which we see here in case of lead food perception by knocking down DRL1, or by having less food intake by the worms in case of E2 mutants. So the less fat storage was still there when we had the SEC1 mutation or the knockdown. In addition to that, we also looked into other metabolic parameters like the ROS levels, autophagy, oxygen consumption rate, which remained unchanged even when we had this p38 mutation, showing that p38 pathway can be placed downstream to metabolic reprogramming. And if it is so true, then it should be able to upregulate the xenobiotic detoxification genes. And indeed, when we looked into the role of SEC1 or the p38 pathway in upregulation, we see that the extent of upregulation that we see under dietary restriction could not happen when we had this mutation in the p38 pathway in most of these panel of xenobiotic genes. And we also phenocopied this result by having a reporter strain of xenobiotic gene CYP35B1 driving the GFP. So there's a higher intensity of GFP under dietary restriction, which actually decreased when we had the mutation in the p38, showing that xenobiotic genes are regulated by p38 pathway during dietary restriction. So if we could place p38 downstream to metabolic reprogramming for the upregulation of xenobiotic detoxification, the question still lies that how is this p38 MAP kinase pathway getting activated? What are the molecules which can lead to the activation of this downstream molecule, which is PMK1? So we had the idea or the hint from the GCMS data that we had of the dietary restricted worms, showing that the polyunsaturated fatty acid molecules like the linoleic acid or the eicosapentaenoic acid, which were enriched among the total lipid profile of the dietary restricted worms, we could see that it was enriched, and thus this PUFA biosynthesis process could be important. So if we abrogate this PUFA biosynthetic process by having a mutation in the FAT2 gene or FAT6, FAT7, then it should not be able to have the beneficial effects. So here we see that when we have this mutation in FAT2 or in FAT6, FAT7, the extension in the lifespan which could happen in DRL1 knockdown could not happen so. So showing that the PUFA genes or the PUFA biosynthetic pathway is required for extension in the deer lifespan. So does that mean that it would also activate the p38 MAP kinase pathway? So yes, we see here that there is a context-dependent activation of phospho-PMK1. For example, you can see here that dietary restriction leads to activation, and that activation goes away when you had a mutation in the PUFA biosynthetic gene FAT2. And the same was replicated or reflected in the activation of the xenobiotic detoxification genes. The genes were, recalcitrant to further upregulate. Some of these genes were recalcitrant to further upregulate upon DRL1 knockdown and having a mutation in the PUFA synthesis genes in FAT2. So showing the context-dependent activation of the V38 pathway and the xenobiotic detoxification genes. So we thought that, okay, fine. So if we could bypass the dietary restriction part, and if we could just supplement these PUFA molecules, will it be able to activate P38 and the xenobiotics and provide the beneficial effects? So yeah, we see here that, upon supplementing the PUFA molecules like EPA or LA, it led to activation of the P38 pathway by having a higher phosphorylated PMK1, and this was reflected in the reporter assay of CYP35B1, which was getting higher as compared to the ethanol, or by giving EPA that was led to increase in the GFP. So if I just go back one slide, I can tell you that, even on abrogating the PUFA synthetic pathway or having a mutation in the FAT2 also led to extension, also led to activation of the P38 pathway. So for that, we thought that if there is a mutation in the FAT2, these molecules which FAT2 works upon, for example, oleic acid, stearic acid, or palmitic acid, these molecules could accumulate and also lead to activation on in P38 MAP kinase pathway. And indeed, we see here that the phosphorylation status of PMK1 is higher when supplemented with OA, SA, or PA, which is also reflected in the xenobiotic detoxification genes by looking into the CYP35B1 reporter strain. So through this, we can say that it is the homeostasis of the fatty acid molecules which is important during DR. So in order to conclude, I would like to say that the role of PUFA molecules, the homeostasis, and the P38 MAP kinase axis is really important in connecting the metabolic reprogramming happening downstream to DRL1 to the cytoprotective response to have a extension in the lifespan. So this study is in revision in Nature.com these days, and I'm working for it. So with that, I would like to conclude and acknowledge my supervisor, Dr. Arnab Mukhopadhyay, for his constant support, and the molecular aging lab members for their support, and the NII core funding, and Professor Jennifer L. Watts for the GCMS data, and Professor Gordon Lithgow for his scientific inputs. Thank you so much, and I welcome your questions. Tumor regenesis. Tumorigenesis. Okay. So I haven't looked into much of the details of the role of PUFA molecules, but I suggest that dietary restriction by itself has a better, it leads to a betterment of the cancerous situation or the tumorigenesis, as in dietary restriction leads to lesser formation of the tumors. So I would assume that, just supplementing these PUFA molecules would also suppress the formation of tumors and would make the organism or the animals healthy. Mhm. Sorry, I didn't get the question. Can you please repeat? Yeah. Could you? Yeah. Your voice is breaking, so yeah, I could not hear the question properly. Time restricted feeding. Um, yeah. So there are actually dietary restriction paradigms are quite variable, and there are various different types of feeding paradigms or regimes that that people have followed, and every type of dietary restriction is has actually proven to give the beneficial effects. So it can be both ways, but I really need to look into that. How could this p38 MAP kinase pathway or the PUFA molecules would be important in the time restricted feeding paradigm? Was I able to clarify? Hello? Hello? Hello?
Hello.
Thank you very much,
Yeah.
Anita. Sorry about that. There was a loose connection this end.
Okay.
Thank you very much. If you could disconnect now, we'll get a move on.
Okay. Thank you so much.
Great
Thank you so
talk.
much for the,
Um,
yeah.
so